Thank you very much, Dr. Yu, for a very nice introduction, and also for the organizer for inviting me to present our study here in this talk. So I would like to start with this quote from Arthur Clarke: "Any sufficiently advanced technology is indistinguishable from magic." So this is the quote that was mentioned or highlighted in this magazine from the Pathologist at the end of 2023. As you can imagine, as generative AI came into our life, exploded into the public consciousness, things have been like magic, right? How we interact with AI these days were unimaginable maybe a few years ago, or even maybe 2 or 3 years ago. So this is what has brought us here today. And for the pathology, the future is really what you see here, the interaction between pathologists and AI. So I'm not pathologist. I am a biomedical scientist slash engineer. But as I interact with these beautiful images and very complicated and complex images, I know one thing for sure is that AI is here not to replace any expert eyes, but to be the tool that is empowering as a partner for pathologists. So in this talk, what I will share is what is really the need for us to have this partnership. And I will talk about it from the perspective of how clinical trial is done and what is the impact of clinical trial. And I will go on into the utility of AI as a tool for pathologists and how it has improved the performance. And of course, the last one is some food for thought and how we can actually further expand AI and how we can make sure that it can impact what we want it to be. So just to put things into a concrete example, I would like to start with this slide on liver disease called MASH, metabolic dysfunction associated steatohepatitis. This is a very common metabolic disease impacting the liver and started by steatosis or accumulation of the lipid in the liver, and which can lead to hepatocyte ballooning, inflammation, and fibrosis. So what is very interesting here is that in clinical trial, liver biopsy is primary endpoint. Liver biopsy is usually a gold standard for evaluating the disease and also for diagnosis of the disease. So as you can see here, it involves very complex system where we have steatosis scoring, ballooning scoring, inflammation, and fibrosis. And in the clinical trial, this means for the clinical trial endpoint, usually a drug will be considered successful if there is a resolution of MASH activities, which is the inflammation 0 and 1 and ballooning 0, and or, depending on which regulation you're using, is one-stage fibrosis regression. So, but what is really the problem here? So this is a publication back in 2011, and then it's again proved true even until now, is that pathologist variability, both intra and inter, is very high. Yes, variability is very high. So concordance between pathologists is very low. For example, here, when we see for steatosis and fibrosis, is relatively substantial agreement we can see here. But for example, lobular inflammation, portal inflammation, and ballooning is relatively low. So this is just the way it is because the nature of the disease is very complex, and then the categorization of the scoring is probably also not completely perfect. Okay, I really like to go back to this paper again and again. This is a very relatively recent paper on highlighting how difficult it is to take a look at feature in this disease. So this is a study where there are 10 pathologists involved, or 9 pathologists involved, to take a look at 10 slides of liver. And then they were asked to actually annotate hepatocyte ballooning. So as you can see here, there are 10 slides, and then for 9 pathologists, and then you can see that the number of balloon cells annotated by this hepatopathologists are very much different. And this one cell here, in one of the liver meetings, international one 2 years ago, this was mentioned as the most famous balloon cells in the world. And do you know why? And this is because out of 2,400 balloon hepatocytes that were annotated collectively by all of these pathologists, there was only one balloon cells that was agreed by all 9 pathologists to be balloon cells. So why is that? I think I will have to refer to our pathologist colleagues, but this is just highlighting how difficult and how complex the problem is. Okay, now why do we even bother with all of this pathologist variability, right? So if we look back into clinical trials, for example, there's this one post hoc study from a clinical trial of Emergence where primary and secondary histologic endpoints were not met, and also there was a high placebo effect rate. This placebo response rate was also high. So it could be that, is it really there is something going on with the trial, or can it be also because the measurement is suboptimal? So in this study, what happened was that we have the inclusion of the subject read by P1, by one pathologist, and then at the end of the trial, it was read by three pathologists. So as a post hoc study, this study was repeated again, so pathologists were asked to read it again. So the results were actually surprising, is that when the same pathologist was asked to read it again, and 1 in 4 patients or 1 in 4 subjects that were initially included into the clinical trials were actually didn't really meet the criteria for inclusion after the second read. And 1 in 6 met primary endpoint, 1 in 8 failed in NASH diagnosis. And if you take a look at the variability between pathologists, actually 14% of the subjects were disagreed by the other pathologists to be included. So then when we are designing clinical trials and then we see the results, and the question, when there's a suboptimal result, the question that comes to mind can also be, are we misclassifying our subjects? Is it because the suboptimal measurement, suboptimal quantification of this disease, then we couldn't get our patients. So FDA recognized this issue. So as I showed you earlier, pathologist variability has been widely recognized. But FDA's opinion is that liver biopsy is still the primary endpoint, but FDA is also open to any advancement in the field, and one of these is the use of AI digital pathology to help and assist pathologists. So, okay, now in the next couple of slides, I will be sharing with you what is the utility of AI digital pathology as a pathologist assistant. So to give you an illustration, I will move away from the liver disease. And this is one of probably the very first few papers that was really looking into the impact of AI digital pathology in impacting the pathologist. So this is in breast cancer. The AI tool, as we have seen here in this Congress and also like outside from our vendors, they're having a very nice visualization to help pathologists in evaluating. And here we see that for micrometastasis, for example, the sensitivity of the detection is higher when there's an assistant by AI. And also this is another paper, also one of the earlier paper on prostate biopsies. And we also see a trend of increase in agreement between pathologists when AI was used. Again, another paper on pathology in, which one is this? This is in the liver cancer. And they were looking extensively into different pathologist cohort from the specialists and also from non-expert. And we also see that, if we see the gray one here, unassisted, and then the purple one assisted, assistance from AI improved the performance. So what about the trend in the field? So if you see that, if you take a look at this website from FDA for the artificial intelligence enabled medical devices, actually out of 1,247 approvals as of last month when I prepared like this one slide, only 6 out of these 1,200 devices that was mentioned are actually for pathologists. And if we take a look at these two are actually from page prostate and Galen second read. And then the context of use is here is really also for software algorithm device to assist users in digital pathology. So the trend is there. And then let's take a look what happens if we go back again to the story of the liver. So digital pathology in the liver already started as well since around 2019 or 2020. And then there were 3, and until now, 3 bigger players in this field. And then from 2020 when it was started and until now 2025, we finally have one platform that's being approved, not by FDA, but by EMR. So I think this is also showing us that the field is moving and responding in the right direction. So we actually have a lot of promise here. So I'm going to discuss a little bit of the approval from EMA for that one that AI digital pathology vendor. And this is just a publication in Nature Medicine, where they actually have this visualization of the different features that I mentioned earlier: statosis, hepatocyte ballooning, inflammation, and fibrosis. And you can see that the platform provides you some visualization of what happens on the slide, and then they use it for pathologist QC and review of the score. And then if the score by the pathologist and the computer by AI agree, then the score is finalized. But if there's a disagreement between this pathologist and AI score, and there will be a second pathologist review coming in, and then see if there is any agreement. If there's no agreement, then there will be a consensus panel. So the proposal is that we can actually help in first screening to have like the first agreement. So how is the result? So this is the comparison for all of the four features. So for steatosis and for fibrosis, there was no inferiority compared to the manual read. So the blue one being AI assisted and the gray one is manual read. And for lobular inflammation and ballooning, we actually have a significantly higher agreement for AI assistance compared to the manual read. So in the next few slides, I'm going to share the study that we have performed. And this is specifically for liver fibrosis. And in here, we are utilizing second harmonic generation. This is the technology where you can just use unstained slides. And when there's photon emitted into the slide, and then there will be a reflection from second harmonic generation signal from the structure that looks like collagen or fibrosis like this. So the platform also has a scoring system from the continuous value as well as from the staging, and also visualization of where this disease in the spectrum of the severity from the F0 to F4. So we had 4 pathologists, all expert hepatopathologists in the study, and we have 120 samples from clinical trial being read, and there are 4 rounds of read, as you can see here, and then the read was alternating between with AI and without AI. And this is a very rigorous study where pathologists have to read 30 slides per week. And then after that, we can compare what is the impact of AI on the pathologist variability. So how do we know that pathologists are really using this? So first of all, we provided survey to the pathologists, and we asked them, what do you think the utility of SSG images and also the score? So this is as what you can see here. We have here read without AI. They are just using H&E and Mason Trichrome. And read with AI assistant. On top of H&E and MT, we are providing SSG images, SSG image, and also the Q fibrosis score. So we asked them, do you think SSG is useful? And yes, most of the pathologists, actually a majority of it, think that SSG is useful, as well as Q fibrosis value. And how do we know is that this usefulness translate into the reliance or that they actually follow what AI tells them? So in here, we compare the fibrosis score and correlating it with the Q fibrosis score. We see that with AI, we have a higher Spearman R, so from 0.69 to 0.82. And you also see that the variability here is relatively lower, and you also have a tighter data spread along this line. So quickly into the result, we have an increase in pathologist concordance in terms of kappa with AI compared to with, and we repeated the study into 2 randomization and 1 and 2, and we also saw the same result. And we also saw that while this is improving the pathologist concordance, the concordance is much better. The increase is actually much higher when you are looking at the early stage of fibrosis. This is where fibrosis is generally very fine and also more difficult and could be more challenging to evaluate. So now we are talking about clinical trial, and I was talking previously about the misclassification of the subject in the clinical trial. And in here, we also saw that if you take a look from this differentiation between inclusion and exclusion. So in clinical trial, what we have is F2, F3 usually is the inclusion criteria, and the exclusion is F1, F0, F1, F4. We see an increase in here. And also, when we are talking about the change, So in the inclusion, while 4 pathologists agreed for 45% of the subject to be in inclusion, to be in the inclusion subjects, after utilizing the AI assistance, we actually see that this inclusion increased to 71%. So the same thing, we have a 45% increase for exclusion, as well as a 24% increase for the treatment evaluation status. I have to say that because this is a retrospective study of a negative study, so there is not much treatment effect that we could see. So I think this contributes to the lower degree of increase for the treatment evaluation. So secondly, because in the clinical trial, FDA also acknowledged that there is this issue with pathologist variability. One way in mitigating this issue is that we can use 2 pathologists, and if there is any disagreement between pathologist A and pathologist B, then we will need to call an adjudicator to finally have a score that everybody can actually agree on. So this is what happens if we simulate it based on the data. So we can have a reduction is almost 25% when we are using AI assistance. So what about the impact on study power? So here, if we take a look at the kappa of 0.4 that usually we have without AI aiding, with the variability that we have between pathologists, the power is actually almost only 40%. While usually in the clinical trial or in a good study, you want to have a study power of 80%, which means that if you want to achieve this with AI aiding, we can actually increase it to 0.7 kappa, and the power is 65%. To further increase it to 80%, it will mean that we need to increase the sample size, so from 600 to 800 or 400 per arms. But this number, if we are not using AI assistance, it means that we really need to increase the sample size to a high degree in a way that it increased from 600 subjects to 1,400 subjects. So you can imagine when we can increase this pathologist variability, it means that we can increase, when we can increase pathologist concordance, it means we can increase study power, and which in turn, we can also actually reduce our sample size, which means that less time and also less cost. Just a quick one on the summarizing what is happening next and what we have to do next. So one of the challenges in AI digital pathology that I've also heard today, both here in the digital pathology section and also up there in the radiology section, is that ground truth or gold standard is always a challenge. We are developing an AI tool based on the ground truth that is not really ground truth. So at the moment, pathologist median is usually used as a ground truth, but how can we actually make it closer to the really to the real truth of the truth? So in this case, we also take a look at the effect of AI aiding. So here, for example, for one pathologist, the correlation with pathologist median is 0.72 without AI assistance. And with AI assistance, we can also see that everything converge into the middle line, which means that hopefully, in this case, we also can help pushing the truth. But the next challenge, of course, if you have expert pathologists in the cohort of the 3 pathologists, and then we have another cohort with, for example, 2 non-expert and 1 expert, or maybe the cohort has all non-expert pathologists, your ground truth, your pathologist median will shift. So there are many ways in approaching this problem. And one of them is from the statistical perspective. I'm not going to talk about this, but the bottom line is usually instead of measuring the pathologist median or measuring the majority voting, you can take into account the reliability of each reader and also use an estimation of what will be the truth of this distribution. Second one, again, this is the question of can we really use AI assistance to close the gap between the median that we will get from the non-expert pathologist and the pathologist, then the pathologist. I don't have an answer now. Probably there's something that the field can move forward, but this will be closer to the reality because let's say clinical trials or anything else, usually we don't really have access always all the time to the expert hepatopathologist. And the next one, what if AI giving you the wrong answer? So what happens? So what happened is in this one study before in 2 more, so it shows that when the model is incorrect actually there's more chance for pathologists to actually lead to also incorrect answer. And this will be related to, are we really relying too much on AI, and how much reliance should we actually have? So that's also another challenge. And this is also from our study. So when we see that, when pathologists think that the AI give you an underscore or an overscoring, then they also tend to not follow it, and then thinking that this is not useful, and it may also lead to confusion. So the key message here is that it is critical for pathologists to know what is the AI tool that they're engaging with, and when it is actually strong, and when it is actually um, less strong or actually need improvement. So it's always in the end, and we always have to go back to the judgment of the pathologist. It's a very last slide. We were all very excited yesterday about the Pat Chat. Um, this is also something that looks like magic. Is it really happening? It is happening. But as if we are always saying that we need to test AI, also the way we test medicine. And I think what is necessary in the field is that all of this excitement, all of this new tool that we have in the field, this is the time for us to really test it, depending on the context of use, if this is actually really delivering and really answering what we need in the field. So closing, I hope I convince you that pathologists and AI can be a strong partner. There are many impacts that can be brought into many things, and then also including the clinical trial that I showed you earlier. But this is not the end of the story. We still have to continue doing our work in order to put the AI in the right context so that we can all like benefit from it. Thank you. I think I'm losing too much time. Thank you.